So today we will start with inborn errors of metabolism. So this will complete the genetics topic, and we will go, if possible, if time permits, we will also start with the evolution. Okay, inborn errors of metabolism are nothing but they are large class of genetic diseases. So that means inborn errors, name itself indicates that by birth they will come. Okay, they are congenital disorders of metabolism. So what is metabolism? Both anabolism and catabolism together it is called as metabolism. So here in the, whether it is in catabolism or anabolism, if there is any problem or any mutation in the genes which are involved in the metabolism, that can result in these metabolic disorders. Okay, so this will result in the accumulation of substrates or products. Okay, so this was actually first, the term inborn errors of metabolism was coined by British physician his Garrod. Okay, so I am not going in depth. Okay, briefly I will explain you. So here, like, what is metabolism? It is mainly for giving the energy. It supplies the energy so to the body, and not only energy, various biomolecules like glucose and other biomolecules will be supplied through the metabolic pathways. So here, if there is any so there are various enzymes which are involved in this metabolic pathways. If there are any defects in the genes which are coding for these enzymes, it results in the accumulation of substrate or product. And usually inborn errors of metabolism are present from the birth, and sometimes the symptoms may appear at the time of birth, and sometimes they appear afterwards in the life. So there are different inborn errors of metabolism, and few of them are fructose intolerance, carbohydrate metabolism disorders, that is galactosemia, maple sugar urine disease, amino acid metabolism disorders like phenylketonuria, fatty acid metabolism disorders, then urea cycle disorders, then organic acidemias, mitochondrial disorders, peroxisomal disorders, and lysosomal disorders. So we will discuss some of the important ones. Okay. So phenylketonuria is the important one. So it is an autosomal recessive metabolic genetic disorder. So it requires to be in the recessive form. Okay. That means 2 copies of the gene should be present, and this is autosomal, present on the autosomal, that is chromosome. Okay. So 2 alleles are required to express the symptoms. If both the parents are carriers of phenylketonuria, then 25% chance of any child having this disorder, and a 50% of chance that child will be carrier, and other 20%, 25% of the child will be healthy. Okay, so this is characterized by homozygous or compound heterozygous mutations in the gene of hepatic enzyme phenylalanine hydroxylase. So in this, the gene encoding this phenylalanine hydroxylase enzyme, so if there is any defect in this enzyme, then it results in phenylketonuria. So it will make that enzyme non-functional, therefore it accumulates the substrate. So this enzyme is required for the conversion of phenylalanine to tyrosine. So when this enzyme will become inactive, phenylalanine will get accumulated. Okay. So, and it gets converted to phenylpyruvate, okay, also known as phenylketone, okay, which is detected in the urine. So, carriers of the single phenylketonuria allele do not exhibit symptoms. So, as it is recessive, 2 copies are required, okay, and it is located, this gene is located on the 12th chromosome at the band Q arm, 22.22 to 24.2, that is the location. So as of 2000, around 400 disease-causing mutations have been found in this gene. And this is the example of allelic genetic heterogeneity. So this is the pathway. So this is blur. So you can check in the textbook. Then alkaptonuria. So this is also a rare inherited genetic disorder which the body cannot possess the amino acid phenylalanine to tyrosine. Okay. So which occurs in the protein. So it is caused in the mutation, that is in the enzyme homogentisate 1,2-dioxygenase. So if a person inherits these abnormal copies of from each parent, the body accumulates an intermediate compound which is homogentisic acid in the blood and tissue. Okay, so this is due to the presence of alkapton, which is oxidized form of homogentisic acid, which will make this urine dark color. So therefore, this is called as alkaptonuria. And this homogentisic acid oxidase is required for the conversion of this homogentisic acid to mallyl acetoacetic acid. So this enzyme is absent. So the homogentisic acid is passed out in the urine, and when it got exposed to the air, it will convert into black color. And this causes damage to cartilage, osteoarthritis, and heart valves, kidney stones, and in other organs also. And your symptoms usually develop in the people over 30 years of age. Okay. So likewise, you have many examples, but as you have many topics to cover, I am just discussing one or two. So here there is a small story behind this alkaptonuria, actually how it got discovered. So there was a scientist who was working as a he was a medical graduate. So he was working as a part-time in 2 hospitals as a nursing. So he used to help the doctors. So in one hospital, when the baby was born, so his diapers or something, so he was nappy, he used to throw that. So when he kept it for some time, when it got exposed to after the urine was passed, so the urine got exposed to air and urine turned into black color. So he was very surprised. So how this has happened? So this is something new. His observation was like he was surprised. Then he used to work in another hospital. So in the another hospitals also, on the same day or 1 or 2 days in the gap, so he observed another baby was born, and same kind of observation was there. So even that baby passed urine, and he used to change the nappies. So again, the urine when it got exposed to air, it turned into black. So then he was like, this is the first time actually, like he didn't observe anything like that. So then he was surprised, and to his like anxiety, he asked whether these two children or the parents of these two children are somewhere related also. Then he found that both the parents are cousins. So then he came to know, okay, because of some genetic problem only, both the babies got this particular condition. Then later when he became scientist, he worked on that, and then he got to know that this is alkaptonuria. Later, more advancements have come and they could discover. Okay. So albinism, goiter. Okay, you can just read it. So just you have to remember is that the enzyme, what is the name of the defective enzyme, and what is the pathway it is involved in, so that if there is any defect, what is the product that is getting, that is either it can be substrate or product which is getting accumulated. Okay. And so what is the genetics behind that? So most types of this inborn errors are inherited in an autosomal recessive fashion. That means both mother and father should be affected, and people have only one affected copy. They don't usually show any symptoms, and they are carriers, as we discussed yesterday. And the symptoms of inborn errors may be according to the specific disorder, because all the disorders are not same metabolic pathways, and severity of mutation also will show you like what kind of symptoms and severities of symptoms. Some they can start with like immediately after the birth, but some may occur after some time, and the symptoms may be from mild to severe. And the symptoms in the infant might be lethargy, means weakness, problems with feeding, low body temperature, vomiting, quick breathing, seizures, and abnormal muscular rigidity. And some additional problems might be failure to grow properly, developmental delay, muscle cramps, problems with nerve pain, movement disorders, behavioral and psychiatric problems, then hearing or visual problems, recurring vomiting and diarrhea, abdominal pain, jaundice and liver problems, enlarged internal organs, heart problems like cardiomyopathy or heart attack, skin rash or abnormal pigmentation and unusual odor. Okay, maple syrup disorder. So in that we can see that the urine, it smells like maple syrup. That's why the name maple syrup urine disorder has come. So what is the treatment? So immediately now you can see that newborn screening, neonatal screening was introduced. Like most of the hospitals, immediately after birth, by urine sample, most of these metabolic disorders will be screened. Okay, so if there is no screening, what happens? We don't know what is a metabolic pathway, and we usually feed the baby with all the food which is available. But if there is such metabolic disorder, the food may be poisonous. It is like a poison because it get accumulated. Suppose phenylketonuria, phenylalanine gets accumulated. If again we are giving the food which is rich in phenylalanine, what happens? It will increase the severity of the disease. So first thing is we have to screen the babies. Then, and the kits are also very cheaper nowadays. It is not much costly. Even we can ask the hospital people also, they will do it. And according to that, we have to supplement the metabolic products that are blocked because of defective genes. If they are blocked, we have to supplement. If they are accumulated, we have to reduce. Reducing metabolic products that are too high level, preventing situations that trigger the symptoms of these inborn errors, and providing symptomatic treatment if they have any seizures. So we have to give anti-seizure medicine. If they have vomitings, we have to give anti-vomiting medicines. Like symptomatic treatment should be given. It cannot be completely eradicated. Okay. So in extreme cases, the children, infants, they cannot live for longer periods, so they will die because of this metabolic disorders. But if we prepare with the proper diet and all these things, they will survive. Then all this has led to one gene, one enzyme theory. So that means the hypothesis was put forward by Beadle and Totem, where a gene controls the structure and functional trait. Okay, so that means a protein or enzyme is coded by a gene. So Beadle and co-workers have found red eye color of Drosophila melanogaster, which is controlled by two genes. Okay, and a piece of larva destined to form the vermilion eye has made to produce red color if it is placed in the body cavity of larva having the red eye. And the later provides its enzyme for brown color. So later, Beadle and Totem, so in Neurospora crassa also, they have found this kind of observation. And finally, they found that there are three types of auxotroph requiring amino acids: ornithine, citrulline, and arginine. Okay, so the phototrophs is nothing but it synthesizes all complex metabolites required for its growth. Okay, so auxotroph is the mutant which is not able to prepare its own metabolite from the raw material. So this auxotroph, it requires ornithine for its growth and do not contain arginine and dies due to protein deficiency. So what happens here? The auxotroph, it requires citrulline, possesses ornithine but no arginine. When citrulline is supplied, the auxotroph comes to have arginine, like that. That means, so here you can see every step we have to provide the particular enzyme and particular substrate. Okay, so here ammonia and sugar, when converted to, when there is enzyme A, it will produce ornithine. When this ornithine is metabolized by the enzyme B, it will produce citrulline. And this citrulline, when metabolized with the enzyme C, it produces arginine. Okay, that's why then the Beadle and Totem has found that each enzyme is encoded by a particular gene, and this each enzyme is metabolizing a particular step. A substrate is getting converted to a product. Okay. Then they got the Nobel Prize in 1958 for their work. Then, so at every step, they tried to block that particular gene. So that step was not able to perform. So that's why they found that each gene, if there is any mutation in the gene, particular enzyme was, so you can see in the wild type, precursor A, precursor is getting converted to ornithine by the action of enzyme A. Ornithine is getting converted to citrulline by action of B. And citrulline is getting converted to arginine by gene C. If there is mutation in gene A, there is no enzyme A. Therefore, this precursor get accumulated, but it won't get converted to ornithine and other steps. And in second step, there is mutation in gene B. Therefore, the first step is normal, but the second step will get blocked. And when there is mutation in gene C, the third step gets blocked, but the first two steps will be normal. So this is nothing but, so by this specific enzyme experiment they have designed, they could find that each gene is encoding a particular enzyme which is involved in a metabolic pathway. Then another concept has come, that is one gene, one polypeptide hypothesis. So it has some drawbacks. This one enzyme, one gene, one enzyme, because all genes doesn't encode enzyme. Okay. So all genes do not produce enzymes in their components. Some of them just control the other genes. Okay. And enzymes are proteinaceous. All enzymes are proteins, but not all proteins are enzymes. And some RNAs also, they have enzymatic activity nowadays. Ribozyme. So whose work was done by Ramakrishnan, Venkatraman Ramakrishnan, Venki Ramakrishnan, other name was. He was of Indian origin and he got Nobel Prize recently in the sense some 10 years back. Okay. So that his work is on ribozyme. So which is RNA enzyme. Okay. And a protein or enzyme molecule may consist of one or more types of polypeptides. Then Yonovsky, he found that enzyme tryptophan synthetase of bacterium consists of two polypeptides. So polypeptide A and polypeptide B. So which is alpha type and beta type. That means each gene is encoding two different polypeptides. Okay. So that is change in one of the two genes causing inactivation of tryptophan synthetase by stopping the synthesis of alpha and beta polypeptide. So here two enzymes are encoding the two forms of same gene. Okay. And here you can see hemoglobin also. It has four polypeptides. And synthesis of two types of polypeptides is controlled by two different genes present on two different chromosomes. Okay, so one gene is encoding one polypeptide, which is a protein. Not all proteins are enzymes, but all enzymes are proteins. Okay, so this completes the genetic topic, genetics. So sorry, I couldn't cover all the topics. So I just gave you the examples, important topics. You can just read the other one. So they are simple, understandable. Okay, we will have just discuss a brief thing about evolution. Okay, so evolution is nothing but change of all forms of life over generation. So evolutionary biology means, so there will be throughout the time, so there will be changes. So there are two theories like survival of the fittest, struggle for existence. So that means an organism, it continuously strive to exist, and that strive for existence and for survival, to become fit to survive under certain conditions. So it requires continuous change, whether it is in the genetic level or morphological level or adaptations, lifestyle, or so and so. So the evolution is a process of change in all forms of life over generation. Okay, whether it can be genetic changes like mutations, okay, then morphological changes, etc. Because genetic changes finally gives rise to the morphological changes, because genes encode the phenotypes. Then theories of evolution. So theory of special creation. So that means initially how life on earth was generated. So it is due to supernatural power, God, Such kind of theories were have come, and all living organisms were created same, and they were created in their present form. Their bodies and organs are fully developed to meet the requirement. So here, this is of personal belief. It is a belief, but we cannot prove. When it comes to science, we cannot prove whether God is there or not. Okay, whether God has created this or not. There are no experiment. You cannot call the God to like prove something. Okay, so this is a type of belief. Okay, that might be true or not, but we cannot prove. Only if we prove, then it is science. So based on that, it is like the limitations are, it is completely belief. No experimental evidence can be given. And age of different fossils are different time. So now we know that first single cell organisms have developed, then multicellular organisms. So all the organisms have not evolved at the same time. So that is one thing. Then second is theory of spontaneous generation. So here also spontaneously they have generated. We don't know, okay, whether it is God or not or whatever it is. But suddenly they have come from somewhere. But even this also cannot be like, there is no logic behind that. Okay, there are several observations like hair of horsetail dipped in the water gives rise to horsehair worm, which is gorgeous. And maggots and flies develop on the rotten decaying matter or meat. So of course, even now, like see, if you keep some food material, although you cover it, you keep it in the fridge also, sometimes they will get spoiled. But that doesn't mean something has spontaneously generated. Might be they are there in the product in some form, but due to the favorable conditions, they started growing. Okay, but here the objections are, this was actually criticized by Lazzaro Spallanzani and Francisco Redi, Louis Pasteur, and all. They have performed different experiments to give that they are not spontaneously generated, but there is some background behind that. There are some other maggots which are present, which developed into the organisms. Okay. Then Lamarckism. So this is called as theory of inheritance of acquired characters. Then after Pasteur, and they have found that it is not spontaneous generation, but because of cell theory and all that, in all cells arise from the cells which are already present. Okay, so like that, after this, he could find that um, he proposed that inheritance of acquired characters, which are defined as the changes developed in the body from the normal characters according to the change of environment. Okay, that means suppose our bodies are like this because our environment is like that. And if we simple like polar bears and bears, you can see polar bears, they are white, their genetic composition is also different because they live in cold conditions. But normal bears which live in hot conditions, their composition is different. So that means our body, it starts modification according to the environmental condition. Because of new needs, the body will change. And disuse of organs, if some organs have we are not using according to the age, so they will disappear. And inheritance of acquired characteristics according to the environment, we have also discussed that environment also will show some kind of impact on the genetics. So that's why, so even the environment also will show some genetic some impact on genetics, and new acquired characters will be developed. Then speciation. So it is nothing but new characters are acquired and transmitted to the next generation, so that the new characters accumulate generation of the generation. Okay. So living organisms, they increase size, produce new organs. Okay. They continue to use the organs more. Okay. And the disused organs will be degenerated. And the acquired characters or modifications are developed by the individuals during their lifetime. Okay. And evidence of Lamarckism is mainly giraffe. So best example is, actually giraffes were small only like other animals, but its neck got became taller as it used the particular organ more and more to get the leaves, to get the access to feed. Okay. Then neo-Lamarckism. So according to this, germ cells may be formed from the somatic cells. Okay. So neo means new again. Recent findings in the field of genetics. So Morgan, Spencer, Bonner, Packard. So many people have supported this. So it is mainly due to they have similar nature of the chromosomes and genetic makeup in two cell lines. Regeneration in earthworm, vegetative propagation of plants, and a part of zygote of human female can develop into a complete baby. So this is all because of genetics. So effect of environment on germ cells through the somatic cells. So I told you like environment effects. So it whenever it affects the germ cells, obviously that will be inherited, and those acquired characters will be passed to the next generation. So germ cells are not immune from the effect of environment. They carry somatic changes to the next progeny, and they are directly affected by the environmental factors. Then finally, Charles Darwin, who was actually he worked more on this evolution. So he stated that every population has built a variation in their characters. Okay. And he has postulated some um, postulates. First is overproduction. So the population tend to multiply geometrically, and the reproductive power will increase as much more than required to maintain their number. Okay. So if the number of progeny increases, they can survive more. Okay. Then constant population, they tend to increase geometrically and the food increases only arithmetically. So as this will increase more and more, the food will be limited. Okay. Therefore, they will change according to the seasonal fluctuation. Their food also will change. Then struggle for existence. So those animals which can fight for food and space and which can reproduce successfully, only they can exist. And the competition between the living organisms for the basic needs like food, space, and meat, that is called struggle. And there are three types: intraspecific, interspecific, and environmental or extraspecific. So, interspecific means within the same species, they'll have limited resources, so they'll fight for that. Two dogs struggling for a piece of meat. Then, intraspecific means between different organisms like a prey and predator. One will try to eat other for its survival. Then environmental or extra specific, it is between the living organisms and environmental factors like drought, floods, earthquakes, and all these who will try to survive, only they can continue. And variations, even the genetic variations, all these will also help in the survival of the organisms. So, natural selection or survival of the fittest, who are fit, who can do all this modification, and only they can continue. And useful variations has to get inherited to the next generation, and this species has to be developed. Okay. So, two theories of Darwinism, theory of evolution, and branching descent and natural selection. So, they were actually successful. So, evidences in favor of Darwinism is that natural selection and artificial selection, there was a close parallelism and resemblance like mimicry and protection coloration. Some animals, they change their color to protect themselves from the predators. Okay. And correlation between the position of nectaries in the flowers and the length of proboscis and the pollination insects. So all these were the evidences. Evidences against Darwinism is inheritance of small variations in the organisms which use only when they fully form, and inheritance of vestigial organs which are of no use now, like appendix in humans, and oversized organs, okay, tusks in elephants, then presence of nectar flowers, sterility of hybrids. They do not differentiate between somatic and germinal variations. did not explain the cause of variations and mode of transmission, and refuted by the Mendel's laws of inheritance. Then, so mutational theory of evolution was actually nothing but the observations are the mutations or variations. They actually help in they are the basis for the evolution. Okay, and it is a discontinuous process and occurs by mutation. So whenever there is a mutation, there is increased, some mutations are advantageous and they are more probably selected by the nature. That is called natural selection. And mutation occurs in different directions and may cause gain or loss of any character. Okay. So neo-Darwinism, it is nothing but new. So there are some extensions. So as I told you, it is almost same like contemporary evolutionary theory. So these chromosomal aberrations, mutations, numerical chromosomal mutations, so all this which we have discussed yesterday. So they all help in the occurring of the new variations which are actually selected by the nature. That is nothing but the natural selection. Then reproductive isolation. So Malthusian competition is nothing but the growth is more and resources are limited. Then variation, that is genetic variation, then mutations. So all these, so those organisms which can compete with other organisms, they are actually, and those have variations and mutations, they are actually selected by the nature. That is called natural selection, which is survival of the fittest. And this mutation can result in Mendelian inheritance and genetic variations. And all these together, they will give the modern synthesis theory, which will give that. So all these postulates, they come together as a modern synthesis theory. Okay. So this completes this topic. So you can just check if you have any questions, because we cannot cover all the topics. So you just check if there are any important topics. So I'm skipping some topics here. I'll try to discuss only the important one. Just a second. So speciation is another important topic. I'll discuss this. So speciation is nothing but the evolutionary process where populations evolve to become distinct species. And this was given by Cook, okay, by splitting the lineages. So here, so this was discussed in the Origin of Species by Charles Darwin. So there are four geometric modes of speciation in nature based on the extent to which the speciation populations are isolated from another. They are allopatric, peripatric, parapatric, and sympatric. So it is induced artificially through nowadays by animal husbandry, agriculture. That means we are trying to produce a new species. Okay. And the sympatric speciation actually can be produced by polyploidy. So I told you like diploid, polyploid yesterday in the class. So this allopatric speciation is nothing but the population splits into two geometrically isolated populations. Okay. And a physical barrier divides the population into smaller units. Smaller groups will be formed and they become different species within the same, like maybe continental drift or something. So they will become small populations and they will become two different populations. And accordingly, because of the nature, there will be changes in the genetics and even that starts to produce the different characteristics. Then parapatric speciation, here it is distributed in the widely spread species. It is continuous distribution, no geographical barrier is there, and individuals of each species may come in contact and cross habitats can be seen here. So there is no geometrical barrier, like they can mate or they can even contact each other. Then stratipatric speciation. So it is categorized by white, and the new species arises sympatrically by chromosomal rearrangement within the geographical range of the parent species. So within the same species, there are some chromosomal variations. So their genetics will change within the same species, same group. Then sympatric speciation is nothing but a type of speciation where freely interbreeding population and have no geometrical uh, isolation, geographical isolation, but within the same area, small pockets like will form small groups like. So in that way, it can form into small groups. Then another one is quantum speciation. So it's a sudden emergence of new groups by saltation, and this is more rapid and sudden. It produces new species and in higher groups. This may be due to environmental change. Okay, so these are the important topics in your syllabus. So you just go through the textbooks. If you have still any doubt, you can just contact me. Okay, thank you.